We've talked about the basics of the lymphatic and immune systems. Now it's time to get into the specific or adaptive immune system. So this little cartoon right here, it's not complete. It only tells part of the story, but for the part that it tells, it's accurate. So that's actually something to come back at for fun. Look, ooh, it's got a scary green thing with teeth. That's awesome. Okay, so remember lymphocytes. Specific immunity depends on lymphocytes. The other four types of white blood cells were involved in the innate immunity, all right, the one that was ready to go the moment mom squirts you out. But the lymphocytes are mostly involved with specific immunity. So T lymphocytes, we nickname them T cells. B lymphocytes, we nickname them B cells. The only lymphocytes that were part of innate immunity were the natural killer cells, all right, but they will help kill things that have been marked by the antibodies of the specific immune system. So there is a tie-in, but specific immunity really depends on T lymphocytes and B lymphocytes. So specific adaptive immunity, how is it different from the non-specific or innate immunity? Well, for one thing, it's systemic. It's not restricted to just one site. So, if you cut yourself and you get an infection right at the site of where you cut yourself, that would be innate immunity. Systemic immunity is all throughout your body. It's going after things everywhere, all right? So, not restricted to the site of the initial infection. And specific fights a very specific pathogen. So, neutrophils, for example, will fight any kind of bacteria. But with T and B cells, there is a specific T or B cell for each specific kind of bacterium. So, for strep throat, for pneumococcus, for, whatever, Pseudomonas, all the different bacteria, there are specific T and B cells for each different kind of bacterium. That's why it's specific. And then memory. So when the body's exposed a second time to the same pathogen, the specific immune system responds more quickly. So you see there in the lower right, the primary immune response. The first time you got sick with something, like, when I was a little kid, I had chicken pox and mumps. It took like a week to get over that. Well, that was the first time. What happens is the specific immune system develops memory, and then the next time that same pathogen tries to invade you, it goes nowhere. It gets wiped out before it even gains a foothold. So memory, when we say that you're immune to something, what we mean is you've established memory for that pathogen, and now that pathogen can't get you anymore. So the forms of specific immunity, we have humoral immunity, which is antibody mediated. Humoral, old-fashioned term, refers to fluids, all right? So this is the immune system of the fluids. So antibodies, remember, are these little tiny Y-shaped things floating through your fluids. This is going to be effective against bacteria, viruses, toxins, and other pathogens because antibodies can grab a hold of practically anything. B lymphocytes differentiate into plasma cells which secrete antibodies. So as we'll see, when the specific immune system is activated, you start cloning your B and T cells, and you clone different types. So one type of B cell that you're going to clone is called a plasma cell, and what it does is it cranks out antibodies. That's its big job. And then B lymphocytes and antibodies circulate in body fluids. So the antibodies, remember, just handcuff. They find a bad kind, they handcuff it. B lymphocytes actually can do what we call receptor-mediated endocytosis. Essentially, they eat the bad guys, and then they serve as APCs. Remember, we mentioned that, antigen-presenting cells. So again, different types of B cells have different roles. Antibodies, again, don't destroy. They simply tag foreign cells or molecules for destruction. And then, as opposed to humoral immunity, we have cellular immunity, cell-mediated immunity. This is effective against infected body cells, cancer cells, and foreign grafts. So now we're looking at T lymphocytes that directly attack foreign cells or diseased host cells, all right? So the two types of immunity then, of specific immunity, humoral and cellular. Humoral, the B cells and the antibodies. Cellular, the T cells. Let's talk about antigens. Specifically, what does it mean? A specific immunity requires recognition of a foreign antigen, and the word antigen is a portmanteau word, like brunch, breakfast and lunch, like a motel, a motor hotel. So an antigen is an antibody generator, antigen. What that means is an antigen is anything that causes your immune system to generate antibodies, all right? So an antigen, Ag, is any molecule that can trigger an immune response. Any molecule or even part of a molecule. So foreign proteins and glycoproteins, foreign complex polysaccharides, foreign lipids, lipoproteins, glycolipids. So anything, basically anything that gets inside of your body that isn't supposed to be there, your body will develop antibodies against it. So we call those things that get into your body antigens because they cause you to generate antibodies. So antigens are large, and usually only certain parts of this large molecule trigger the immune response. We have what's called the epitope. So we'll talk about that more on the next slide. But remember, you've seen this before on the red blood cells. You had antigens. So if you had type A blood, that means you had the A antigen. Type B, you had the B antigen, and so on. So we've seen antigens before. So there are foreign antigens, and you also have self-antigens. So one of the things that happens during the training part of the specific immune system is you have to train your immune system not to respond to self-antigens. What do you suppose would happen if you responded to self-antigens? If you started attacking antigens within your own body? Yeah, I'll bet you've heard of that before. Those are called autoimmune disorders, where your immune system attacks your own body. Okay, so antigens and epitopes. So here we see an antigen, all right, that whole big tennis ball-like thing. And then each antigen can have several epitopes. So the epitopes here are the cones and the cylinders and the whatever those other things are. So this particular antigen, this might be polio, it might be measles, it might be coronavirus, it might be anything. But the antigen can have different epitopes on it as well. And when you look at that lower right, what you see is that an antigen can have several different epitopes. Well, you can have antibodies for each epitope. So this antigen, for example, will have 4 antibodies that can attack it because it has 4 different epitopes on it. All right? A lot of versatility in your immune system. All right? You can generate antibodies, a variety of antibodies that will attack the antigen in many ways. That gives you a better chance of getting it. So antigen receptors, we have like at least 1 billion unique lymphocytes in our body. Think about it, 1 billion, that's an awful lot. So again, you have a unique lymphocyte for measles, polio, chickenpox, the mumps, and on and on and on and on. A different one for each one. That's pretty incredible. So each lymphocyte displays only one type of antigen receptor, but that lymphocyte has hundreds that are all the same. So you see those B cells down in the lower left. They're showing, just 4 because, it's an illustration. You can't make it realistic. So, but each one of those B cells, notice it has a different type of antigen receptor on it. One has the blue ones, the other has the green ones. So in other words, those would be like, one would be for measles, another would be for chickenpox, and so on. They have lots of receptors all over the cell. But each one of those receptors is only for one particular type of antigen. And then every lymphocyte spends its entire life wandering through your body, just waiting to come into contact with its specific epitope. It's been trained. Remember we said that these lymphocytes have to go through training. They have to go to special ops school. So during training, they get their antigen receptor inserted. And that from that moment on, they spend their entire time just looking for that particular epitope on that particular antigen. So antigen receptors are inserted in lymphocyte membranes before B cells leave the red bone marrow. Remember, that's where the B cells do their special ops training. And before T cells leave the thymus. That's where the T cells do their special ops training. And so antigen receptors can recognize even artificial molecules that don't exist. We'll talk more about that later. Your body, think about when you shuffle a deck of cards. You're just taking the cards, shuffling them, putting them into a random order. Well, that's kind of what your immune system does. It generates random receptors. And so as it turns out, A lot of those receptors will go after things that we actually know about: polio, measles, chickenpox, hepatitis, and so on. But other receptors have just been randomly generated. They go after things that might not even exist. Or it might be like if we ever had like something from Mars came to Earth and there was a Martian virus, you might already have antigen receptors for Martian viruses just because your immune system randomly generated like a billion of these things. And so you've got them even for things that don't exist, even for weird things like Martian viruses. So notice in the lower right, we're seeing the specific receptors. So you see the antigens, and then you see what it says marker molecule. Those would be the epitopes. So notice how some antibodies can grab one type of epitope, whereas other antibodies grab other kinds of epitopes. And any particular antigen can have different kinds of epitopes. See, so isn't that cool? You've got all these antibodies, and the Ys on the B cells in the lower left are essentially the same as antibodies. So they are ready to grab any specific thing that they've been trained to grab, all right? So major histocompatibility complexes, MHCs, this is a big deal. So proteins are manufactured inside of your cell. Remember, that's one of the main jobs your cells do. They're just big, giant protein factories. Remember, the mRNA and the transfer RNA and the ribosomes. You have the DNA. Cells make proteins like crazy. That's what they do. Cells spend their lives making proteins. Healthy cells make normal proteins, proteins that you're supposed to have in your body. Well, disease cells will make abnormal proteins, proteins that normally should not show up in your body. So what will happen is phagocytes, like APCs, like dendritic cells, like macrophages, like B cells, they'll ingest foreign cells and molecules. And then the major histocompatibility complex molecule takes proteins made inside the cell, or fragments of phagocytized cells and molecules, and displays them on the surface, all right? So every cell in your body will do this, okay? So it's got proteins inside of it. It will take parts of these proteins, fragments, and display them on the surface. And it uses a major histocompatibility complex protein to do that. So MHC1 molecules are found on virtually every cell in the body. We often say they're found in every nucleated cell. Why do we say that? Well, think about it. Red blood cells, remember, don't have nuclei anymore. So every nucleated cell has an MHC1 complex. And what that's going to do is, so every cell in your body is going to take proteins that it's making inside of itself, and it's going to put those on the MHC1 molecules. Well, as we'll see later, what's going to happen is, for example, a T cell is going to come by, and it's going to inspect the MHC1 molecule on the cells of your body. And if it sees a normal cell protein on that MHC1, then it'll just say, hey, nice to see you, how you doing, how's the kids? But if it sees an abnormal protein on the MHC1 complex, that means the cell has been infected. So what do you suppose this T cell does to it? Yeah, it kills it, all right? So that's how your immune system knows whether cells are healthy or whether they're diseased. Your immune system is going to inspect the MHC1. If it sees a normal protein, it'll just let it be. But if it sees an abnormal protein, that means it has to die, all right? Then there are also MHC2 molecules found only on antigen-presenting cells. So only APCs have MHC2s. And the reason APCs have those is, remember, what's the job of the APC? The job of the APC is to ingest bad guys and then alert the immune system that certain bad guys are here. Well, how does it alert the immune system? That's what the MHC2s are for. And we're going to see more about that coming up. But APCs are the only ones that have MHC2s. That's how they tell your immune system that bad guys are here. Whereas, again, the MHC1s are found on every nucleated cell in your body, and that's how other cells of your immune system can tell whether that cell is healthy or whether it's been infected. Okay, so antigen-presenting cells, which I've been talking about, we've talked about before. They include macrophages, dendritic cells, they used to call them Langerhans cells, and B cells. So APCs eat antigens and present their epitopes to the lymphocytes. So APCs have the MHC2, and that's the purpose of the MHC2, all right? It's going to present that. So look in the lower right-hand diagram. You've got an APC, all right, and then there's a helper T cell and a CD8 T cell. The CD8 T cell we're going to call the cytotoxic T cell. It's the killer, okay? And what's going to happen here, just to preview, this is going to be coming up later, but I'll preview it right now. Notice how the APC is using its MHC2 to show what it ate to the helper T cell. Now, if it ate something normal, ate something harmless, then the helper T cell just won't care. But if it ate a pathogen, then the helper T cell is going to go, oh my God, we have invaders in the body. And then it's going to be the helper T cell that activates the killer T cell. And how does it do that? By secreting little proteins called interleukins. There, see, IL-2, all right? So this is a process we're going to see over and over again, so get used to this. APCs eat something, all right? They either eat good guys or bad guys. Whatever they eat, they show to the helper T cell. If they've eaten a bad guy, which means we've been invaded by something, then the helper T cell activates the cytotoxic T cell. Killer T cells are really dangerous, all right? So we don't want to activate them unless we know we've been invaded, and that's what the helper T cell does. So APCs use the MHC2 to display antigens to T cells. If an APC displays a self-antigen, the T cell ignores it, all right? Again, if it's a normal molecule the cells of your body should be making, the helper T cell doesn't care. But if it's a foreign antigen, meaning we've been invaded, then the helper T cell alerts a cytotoxic T cell and says, get to work, bad guys are here, load your weapon, get out there, get them, all right? And lymphocytes tell other lymphocytes about invaders using cytokines such as interleukins. Cytokines are small messenger molecules, and in particular, white blood cells use a cytokine called an interleukin. Inter, between, leukin, leukocyte, white blood cell, interleukins. These are chemicals that allow white blood cells to talk to each other, okay? Interleukins are heavily involved in the coronavirus and COVID-19, all right? We'll talk more about that. So there you see, all right, there's an APC. Great diagram here, all right? Look, numbered steps for you. Oh my God, look at how easy they make it. But there you see what's going on. It's eaten an antigen, all right? And then notice it basically digests it and then breaks it down into recyclable components and voids those. I mean, those might be little amino acids and stuff that your body can use. Why not? I mean, the bad guys got usable parts. You know, if you find a dead soldier, you know, take their ammo, take their gun, take whatever they got that's useful, all right? But then notice that the APC, it's taking the epitope and displaying it on the MHC protein. What kind of MHC would that be here, since this is an APC2? I mean, it's an APC. I gave it away. That would be an MHC2, all right? And then here you see just a different diagram, just a different artist's, you know, representation of this. Here you see the antigen, again, being phagocytized. And then, you know, we go ahead and we take that epitope and we display it on that MHC2, okay? And then the rest of the immune system, the helper T cells will be able to see it, okay? Which type of cell does not have an MHC2 complex? Macrophage, T cell, B cell, dendritic cell, or APC? Which does not have an MHC2 complex? Yeah, that'd be T cells. Only APCs have MHC2s.